What's up, guys?
How's it going?
How was your break?
Fantastic.
Yeah, was it?
Yeah.
Short. I understand that, I know. So before we get into second semester material, I do kind of want to look back as to where we just came from. All the material that we did in first semester, succession and food pyramids and all that stuff. And I think it makes sense to look at that stuff before we move on to new things.
What exactly do you have in mind? Like a test or what? Come on, not a test the first day back, man.
No, not a test. Tell you what.
Not throwing away what I have taught. First semester of biology cannot be forgot, caught as an afterthought. Yeah, it's a lot, so we ought to test and see what you got. Take a shot at a lesson in ecology. 10% of each trophic level's energy. Pass from bottom to the top pyramidically. Oddities to V's, passing ATP up to the carnives. Now biology, levels keep it orderly. Beck-Poot comma keeps them deeply in my memory. If a population is racing towards infinity, there's a limit within it called carrying capacity. Population density tells a different story. The inventory of an organism in a territory. Births and deaths take a toll, and when they're migratory, INEX succession's our next category. Lichens, moss, grasses, shrubs, coniferous trees, deciduous, now that's primary. Starting on rock, unlike secondary. Soils on the ground, so lichens are unnecessary. Switching gears, it's clear that in our biosphere, biogeochemical cycles are players to revere. Water is recycled from the ground up to the atmosphere, and carbon's by far been the one giving biologists fear. Respiration, combustion, decomposition. CO2's emission condition puts us in a tough position. Plants' mission is in polar opposition. Carbon to a carb is the photosynthesis transition. See, I'm not throwing away what I have taught. First semester of biology cannot be forgot, caught as an afterthought. Yeah, it's a lot, so we ought to test and see what you got. See, I'm not hesitating on information regurgitation, like nitrogen's dependence on bacteria for fixation. Ammonia, nitrates, nitrites, formation, denitrification takes them back to their origination. But I'm not through, cause we've got phosphorus to do. Rock is the starting place, erosion leads to phosphate, and we drink it in the water so that you and I can make DNA and RNA, and now you and I can say that we're through. But see, that's not exactly true, cause how do species like Japanese kudzu impact native ones? They outcompete them for the sun's the non-native. Invasive took over all the native and they overgrew. And ecology, that's your conclusion, cause we got lots to get to, like cells and diffusion, which can be confusing if you don't know the rules of the cell's tools, the mules, the macromolecules, and they're quite the crew. But with them we've got to go easy. They don't always stick like easy cheesy. They make up all that's in the cell, including every organelle. So here they come, the rules of thumb. We'll beat them like a bass drum. Here's their cue. Component of membranes, CH2 groups bound together in long chains. Lipid is my name, long-term energy, the gain, it's insane. The disdain I maintain with rain, flush it down the drain. Check out my fatty acids, drawn as zigzags in fats and waxes. And what if you're wondering if lipids are present? Sudan can do it, brown bag a conduit for life to pass on through it. Oh, I'm sorry, am I getting excited? I must have eaten carbs, my energy's heightened. They're made of CHO with a 1-to-1 ratio. They end with dose of glucose, cellulose. See, I'm not throwing away what I have taught. The first semester of biology cannot be forgot, caught as an afterthought. Yeah, it's a lot, so we ought to test and see what you got. Carb simplified is the saccharide, each with 5 or 6 sides. In a line combined, you'll find it when you bind it with iodine, yellow to black. I feel it's safe for you to predict. Sugar blue to orange when heated up with some Benedicts. Next test, third guest, guess who it is? 3D peptide bound chain of amino acids. Brick of the cell, serve as mail unless they're enzymes clearly defined by the public defeat acronym of mine. Your desk is wet and you don't know what it's from. Throw on some Bioret if violet's made of charm. Last one's a fast one. NAs, nucleotides are certified to provide genetic information. If DNA wants total protection, nucleus impervious to lysosome action, I vote to promote the cell known as a eukaryote, cause the prokaryote forces its DNA to float. By the way, eucs have organelles too, wrapped in lipids that I mentioned in verse 2. Produced by the ER, the smooth but the RER caviar. Here, look at the CR, the ribosomes, the synthesizers of proteins. And Golgi's done got a vendetta against those things. Takes them, remakes them, and the proteins are banished. And Golgi just waves and watches them vanish through the membrane. Reminds me of diffusion, where molecules pass without ATP inclusion. Facilitated says, hey, say you can't help your big size. So my carrier protein will put you on the low side. With concentration gradient, the problem is radiant. You know the sides aren't equal, so there's no point in saying it. Water can move when the membrane approves it, but we gotta know the direction that osmosis moves it. Low solute, we call you a hypotonic solution, where the solution's been subjected to solute dilution. Water moves towards where it's low. So in a hypo, the cell drinks, but in a hyper, it shrinks. But there's another guy who oversupplies the high as he robs the poor side. Energizes ATP is supplied, exemplified as endo and exocytosis. The diagnosis is active, the opposition to passive. But next I want to mention this, thank God for photosynthesis. From chloroplast make glucose in the gas that permits us to last. Our exhalation plus hydration, solar radiation, their creation sets the stage for respiration. See, I am not throwing away what I have taught. First semester of biology cannot be forgot, caught as an afterthought. Yeah, it's a lot, and we ought to test to see what you got. Breathe deep so you can keep breaking down glucose aerobically. Krebs cycle ETC, popping out ATP like candy. All the reason to stay breathing, and if that's ceasing, lactate will be releasing, NAD increasing, anaerobic, toxophobic, pure autotrophic, ethanol's the final straw when oxygen levels fall. First semester's been learned, no stone's been left unturned. Hope I have not thrown away my shot.